In 2020, the Nobel Prize in Chemistry went to two scientists for a discovery that started with an obscure question about how bacteria fight off viruses. Jennifer Doudna and Emmanuelle Charpentier's answer became the reason a child born with sickle cell disease today has a genuinely curative option that didn't exist a decade ago. That's where we're starting and where we're going to end. Define it precisely, because this is a favorite exam distinction. CRISPR clustered regularly interspaced short palindromic repeats is the bacterial genomic locus that stores fragments of past viral DNA. Cas9 is the nuclease enzyme. CRISPR-Cas9 as a technology repurposes this natural bacterial adaptive immune system into a programmable tool that cuts DNA at a site you choose. As of this recording, there are well over 200 active clinical trials worldwide using CRISPR-based therapeutics, spanning hemoglobinopathies, inherited blindness, certain cancers, and cardiovascular targets. This is no longer a bench technique, it's an active clinical pipeline you will encounter in practice. The timeline matters for context. Repetitive bacterial DNA sequences were first noted in 1987 with no known function. In 2012, Jiankui, Charpentier, and Doudna published the mechanism showing Cas9 could be programmed with a single guide RNA. By 2020, that work won the Nobel Prize. And in December 2023, the FDA approved the first CRISPR-based therapy for a genetic disease, a remarkably short path from basic science to bedside. Whatever the editing tool, the target is always the same double helix, two antiparallel strands held by complementary base pairing. Keep this structure in mind, because every editing strategy we cover today is really just a different way of finding, cutting, or chemically converting one specific base pair within 3 billion of them. A myth worth killing early, because it shows up constantly in lay press coverage. That CRISPR is a simple, universal find and fix tool ready to cure any genetic disease. The fact is CRISPR is highly effective for a narrow category of conditions, typically loss of function, single gene, blood, or accessible tissue disorders, and remains technically limited for polygenic disease, in vivo delivery to most organs, and precise large sequence correction. Genome editing didn't start with CRISPR, it's worth knowing the lineage. Zinc finger nucleases and talons both used engineered proteins to recognize DNA sequence, which meant redesigning a protein for every new target slow and expensive. CRISPR's innovation was replacing that protein recognition step with RNA-DNA base pairing, which you can simply reprogram by changing a 20-nucleotide sequence. High-yield framing for the rest of this video. Every CRISPR-based technology is defined by three variables: how it finds its target, whether it cuts both DNA strands, and how the cell repairs or converts that site. Master those three variables, and every tool we cover will make sense. Walk through the sequence exactly as it happens in the cell. A synthetic single-guide RNA is designed to be complementary to your target DNA sequence. Cas9 protein binds this guide RNA. The complex scans genomic DNA until it finds a PAM sequence immediately adjacent to a complementary target. Only then does Cas9 unwind the DNA, check for base pairing complementarity, and if it matches, cleave both strands. High-yield fact, tested constantly. For the most common CRISPR system, SP-Cas9, the PAM is NGG, meaning any base followed by two guanines. No PM, no cutting, full stop, regardless of how perfect your guide RNA's complementarity is. This is a favorite "why didn't editing work here" exam trap. Structurally, think of the Cas9 guide RNA-DNA complex as a ternary machine. Cas9 provides the scaffold and the two nuclease domains. The guide RNA provides sequence specificity, and the unwound target DNA strand pairs with the guide to trigger the conformational change that activates cleavage. Once Cas9 cuts, the cell's own DNA repair machinery decides what the edit actually looks like, and this is the single most tested concept in this entire topic. Non-homologous end joining directly rejoins the cut ends and is error-prone, producing small insertions or deletions that typically knock out the gene. Homology-directed repair uses a supplied donor template to copy in a precise sequence, but it only operates efficiently during S and G2 phase. Compare the two pathways side by side and the clinical implication becomes obvious. If your goal is simply to disrupt a gene, like the BCL11A enhancer in sickle cell therapy, you want NHEJ, and it's efficient in any dividing or even non-dividing cell. If your goal is to correct a specific point mutation, you need HDR, and you're fighting a much lower efficiency ceiling. This efficiency gap isn't subtle. In most therapeutically relevant cell types, NHEJ-mediated editing routinely exceeds 70%, while HDR-mediated precise correction typically struggles to clear single digits without additional engineering tricks, which is exactly why every currently approved CRISPR therapy relies on NHEJ-based disruption, not HDR-based correction. Critical safety concept and a guaranteed exam theme. Cas9 can cut at genomic sites that resemble but don't exactly match the guide RNA sequence off-target effects. An unintended cut in or near a tumor suppressor gene is the theoretical worst-case scenario, which is why every clinical-grade guide RNA undergoes extensive off-target screening before it ever reaches a patient. Off-target risk isn't uniform across the genome, it clusters wherever a near-match sequence happens to exist, which is exactly why techniques like GuideSeq and DiGenome-seq are run empirically for every clinical guide RNA rather than relying on computational prediction alone. A quick mnemonic to lock in the whole targeting sequence before we move on to the next generation tools. The single biggest limitation of classic CRISPR-Cas9 is that double-strand breaks are inherently risky. They can trigger large deletions, chromosomal rearrangements, or activate p53-mediated cell death. Base editing and prime editing were both developed specifically to make precise edits without ever cutting both DNA strands. Base editing works through a genuinely elegant piece of enzymology, first described by David Liu's group in 2016. A catalytically impaired Cas9, a nickase, cutting only one strand is fused to a deaminase enzyme. The deaminase chemically converts a target cytosine to uracil, which the cell's own repair machinery then reads and fixes as thymine. No double-strand break is ever created. Line all three tools up against the same criteria and the trade-offs become a clean exam table. Nuclease editing is the most versatile but the least precise at the single base level. Base editing is extremely precise but limited to specific base conversions. Prime editing sits in between, handling small insertions and deletions that base editing simply cannot do. Profile prime editing honestly against the criteria that matter clinically, and you see why it's called a search and replace tool. Outstanding versatility and precision at the cost of a larger genetic cargo that makes delivery genuinely harder. Prime editing's mechanism is worth walking through once slowly, because it's genuinely a different logic from base editing. A prime editing guide RNA, or pegRNA, both directs the Cas9 nickase to the target site and carries the template for the new sequence. Reverse transcriptase, fused to the nickase, uses that template to directly write the desired edit into the genome. The guide RNA isn't just an address, it's the blueprint. A myth worth correcting directly because it's a common exam distractor. That base editing, since it avoids a double-strand break, is essentially risk-free. The fact is base editors can still cause off-target DNA and even RNA edits, and bystander editing, converting nearby cytosines within the same editing window that you didn't intend to touch remains a real limitation. Worth remembering as a single high-yield line, because it captures why these tools were developed at all. Nuclease editing measures success in percent edited alleles. Base and prime editing increasingly measure success in fold reduction of unwanted byproducts, because the ceiling on precision, not on activity, is now the limiting factor. Carry this comparison forward as one sentence you could write on an exam. Choose nuclease editing to disrupt a gene, base editing to correct a single base, and prime editing when you need a small precise insertion or deletion without a donor template. None of this mechanism matters if the editing machinery never reaches the target cell. Adeno-associated virus, AAV, is the workhorse vector for in vivo delivery. It doesn't integrate into the genome and provokes a relatively mild immune response, but its small packaging capacity is a real constraint for larger cargo like prime editors. Broadly, delivery splits into two clinical strategies with very different risk profiles. Ex vivo editing removes a patient's own cells, edits them in a controlled lab setting, verifies the edit, then reinfuses them. This is how exagamglogene autotemcel works. In vivo editing delivers the editing machinery directly into the body, which is technically harder to control but avoids the toxicity of a conditioning regimen. Delivery is an all-or-nothing editing. Efficiency climbs with vector dose up to a point, then plateaus while toxicity keeps rising. Finding that therapeutic window is exactly the kind of dose-finding problem a Phase 1 trial exists to solve. The adverse effect profile differs meaningfully by delivery route, and this is genuinely testable. Ex vivo strategies carry the toxicity of the myeloablative conditioning regimen itself, while in vivo strategies carry vector-related immune and hepatic risks, plus the theoretical risk of germline transmission if the vector reaches gonadal tissue. For ex vivo cell therapy manufacturing, think of it as a funnel with real attrition at every step. Stem cells are mobilized and collected, electroporated with the Cas9 guide RNA ribonucleoprotein. The edit is verified, and only the successfully edited, viable cell product is released for infusion back into the patient. A critical, testable safety point. Some patients already carry pre-existing antibodies against Cas9 protein or against the AAV serotype being used, typically from prior natural exposure. This isn't hypothetical, it's a standard pre-treatment screening step precisely because it can neutralize the therapy before it ever reaches the target cell. One-line comparison worth memorizing for each delivery platform's defining trade-off. The exam-ready synthesis. The editing tool determines what change is made, but the delivery platform determines whether it's even possible and which toxicities you need to counsel the patient about. A 19-year-old woman with known homozygous sickle cell disease presents to clinic. She has had 4 vaso-occlusive crises requiring hospitalization in the past year despite hydroxyurea therapy. She asks whether she's a candidate for the new gene therapy she read about. This is exactly the conversation exagamglogene autotemcel, brand name Casgevy, was designed for. Quick pause. If this breakdown is helping you, take a second to hit like and subscribe. It genuinely takes a lot of effort to make videos like this, and your support keeps them coming. This is the mechanism worth knowing cold. Sickle cell disease is caused by a single glutamate to valine substitution in beta-globin, which causes deoxygenated hemoglobin S to polymerize and deform the red cell. Exa-cel doesn't touch that mutation at all. Instead, it disrupts the BCL11A erythroid enhancer, a gene that normally silences fetal hemoglobin after birth. Silencing the silencer reactivates fetal hemoglobin, which doesn't sickle and dilutes the pathologic HbS. 2 approved or late-stage Crispr therapies define the current clinical landscape, and each targets a different tissue with a different delivery strategy, a favorite way for exam questions to test whether you actually understand the mechanism rather than just the brand name. Before any editing happens, the patient goes through busulfan myeloablative conditioning, the same regimen used in allogeneic transplant, and its toxicity is a required counseling point, not an afterthought. The outcome data is genuinely striking and worth quoting precisely. In the pivotal trial, the large majority of treated patients were free of vaso-occlusive crises for at least 12 consecutive months post-infusion, durable enough that this is now considered a functionally curative, one-time intervention rather than a chronic therapy. Not every patient with sickle cell disease is an immediate candidate. Eligibility follows a structured pathway that a student should be able to reconstruct. Confirm diagnosis and severity criteria, assess fitness for myeloablative conditioning, mobilize and collect adequate CD34+ cells, then proceed to ex vivo editing and reinfusion only once the edit is verified. Follow the lab trajectory after infusion, and it tells the whole clinical story in 2 numbers. HbS falls as edited fetal hemoglobin production rises, typically stabilizing by 3 to 6 months post-infusion. Delivering this kind of therapy takes a genuinely multidisciplinary team, and it's worth knowing who's actually involved before you refer a patient. One mnemonic to hold the whole Exa-cel mechanism in your head under exam pressure. The Exa-cel pivotal program is a clean example of the standard gene therapy trial structure, worth knowing phase by phase. Early phase work established the editing and manufacturing approach, and the pivotal CLIMB trials generated the efficacy and safety data that ultimately supported FDA approval in December 2023. Plot VOC-free survival over time, and the curve is about as favorable as gene therapy data gets. The overwhelming majority of treated patients remain free of vaso-occlusive crises well past the 12-month primary endpoint, with the curve staying essentially flat rather than showing the gradual attrition you'd expect from a non-curative therapy. Follow patients through the trial's consort flow and note where attrition actually happens. It's concentrated at stem cell mobilization and manufacturing, not at the editing or infusion step itself, a detail that matters for counseling patients about what can still go wrong before treatment even begins. Structured as a PICO question, the way you'd be expected to frame it on rounds. In patients with severe sickle cell disease, does exacel versus standard care reduce vaso-occlusive crises? The evidence, while still based on relatively small single-arm cohorts, was strong enough to support a landmark approval grade of recommendation, reflects genuine efficacy tempered by limited long-term follow-up. Where does this evidence actually sit on the hierarchy? Above case reports and expert opinion, but still below a large randomized controlled trial and honest, testable placement, given that single-arm cohort data, however striking, isn't the same as head-to-head randomized comparison. Compare relative effect sizes across the handful of Crispr gene therapy programs with published data, and the consistency across independent programs targeting different diseases is itself part of what built regulatory confidence in the platform, not just any single trial. Read these trials critically, not just favorably. That's what separates a student who can cite a result from one who can interpret it. Small single-arm cohorts, historically controlled comparisons rather than randomization, industry sponsorship, and short follow-up relative to a claimed lifetime cure are all real limitations worth naming explicitly. The evidence-based synthesis. The data is genuinely strong for what it is, but strong single-arm cohort data and proven lifetime cure are not the same claim. Know the difference, because you'll be asked to defend it. In November 2018, a Chinese scientist named He Jiankui announced he had created the world's first germline-edited babies, twin girls, whose CCR5 gene had been edited using CRISPR, intended to confer HIV resistance, before they were even born. The global scientific community's reaction was near unanimous condemnation, and he was later sentenced to prison. This single case reshaped how the entire field talks about ethics. The timing of an edit is exactly what separates a routine, defensible therapy from a global scandal. Edit a differentiated somatic cell, like a patient's own bone marrow stem cells, and the change dies with that patient. Edit a single-cell embryo before its first division, and every descendant cell, including future sperm or eggs, carries that change permanently. Run the He Jiankui case, or any proposed germline editing, through the 4 classic bioethics pillars, and the tension becomes concrete rather than abstract. This is exactly the kind of structured reasoning an ethics exam question expects. Even routine somatic gene therapy raises consent complexities worth naming explicitly. Patients are consenting to an irreversible, one-time intervention with a relatively short track record, often while facing a serious, quality-of-life limiting disease that itself pressures the decision. The regulatory response to germline editing has been fast and largely unified, worth knowing as landmark reference points. A 2015 international summit called for a moratorium on clinical germline editing. The 2017 National Academy's report reinforced extreme caution, and He Jiankui's prosecution in China sent an unambiguous signal that this line will be enforced. Access is the other ethical fault line that's easy to overlook once you're focused on mechanism. Exacel's U.S. list price is roughly $2.2 million per patient, and it requires a specialized transplant center, meaning a curative therapy exists for a disease that disproportionately affects populations with the least access to it. Zoom out to the global burden, and the equity problem becomes stark. The overwhelming majority of the world's sickle cell disease burden falls in sub-Saharan Africa and parts of India, regions where a $2 million specialized center therapy is functionally unreachable for nearly everyone who has the disease. One sentence worth remembering past this video, because it's the tension the entire field is currently sitting inside. Hold both truths at once. The scientific achievement is real, and the ethical and access questions it raises are not solved by the mechanism working. Somatic therapy for serious monogenic disease is broadly accepted. Germline editing of embryos remains, by near global consensus, a line not yet crossed. Picture the 3 core tools as overlapping circles rather than a strict hierarchy. All 3 can produce a stable genomic change, but only classic CRISPR-Cas9 reliably knocks a gene out, and only prime editing can currently insert or delete a small precise sequence without a donor template. 4 numbers worth having cold before any exam on this topic. Where this field is heading next. briefly worth knowing as background, even though it's less exam-tested than the mechanism itself. Zoom all the way out, and every thread from this masterclass connects back to the same handful of nodes: mechanism, precision, delivery, evidence, and ethics, all feeding into where the field goes from here. Run through this checklist mentally before any exam question on Crispr. If you can answer all 5 without hesitation, you're in good shape. Let's run back through the 3 questions most likely to actually appear on an exam. One line to leave you with, the kind an examiner loves to hear back verbatim. Final synthesis for your notes. Know the mechanism cold. Know exactly what exocell does and doesn't correct, and know precisely where the somatic germline ethical line sits. Those 3 things will carry you through almost any question this topic can generate. Here are the key references and sources for this video. Thanks so much for watching. If this helped, like the video and subscribe for more clear, simple explainers, and I'll see you in the next one.